So Nat5, Cell Biology video number 2, DNA and Proteins. Firstly, we look at DNA and the production of proteins. DNA contains our genetic information and it consists of a great many genes. A gene is a section of DNA which codes for a particular protein. And all these genes make up our genome. DNA is found wrapped around supporting proteins which makes up chromosomes. If we briefly look at the structure of DNA, DNA is a double-stranded helix held together by complementary base pairs. These complementary base pairs are adenine, which always binds with thymine, and cytosine, which always links with guanine. And this is the base pairing rule. Every 3 bases in the DNA sequence codes for 1 amino acid. The DNA base sequence determines the sequence of amino acids in a protein, and therefore the sequence of amino acids determines the structure and function of a protein. Messenger RNA, or mRNA, is a molecule which carries a complementary copy of the genetic code from the DNA in the nucleus to a ribosome where the protein is assembled from amino acids. Now, for this to occur, firstly, the strands of the DNA molecule separate, exposing the bases on one strand. A molecule of messenger RNA is then synthesized using the DNA strand as a template. This messenger RNA now leaves the nucleus for the cytoplasm. As we have already ascertained, each group of 3 bases on the mRNA codes for a single amino acid. The mRNA attaches to a ribosome, and the mRNA is used to construct a molecule of protein. Now, if we have a look at those proteins themselves and the variety and functions of proteins, the variety and function of proteins depends on the sequences of amino acids and the shape of the protein molecule that is produced. The different functions of proteins include structural proteins, hormones, antibodies, receptors, and enzymes. We find structural proteins within the cell membranes and in places like the skin, the hair, and the nails, such as fibrous proteins like collagen or keratin. Hormones now are chemical messengers in the body. They're made of protein, generally transported in the bloodstream. They regulate processes such as growth and metabolism. And examples of hormones includes, of course, insulin, human growth hormone, testosterone, and antidiuretic hormone. Next, we see antibodies. These are Y-shaped proteins. They're protein molecule made by the white cells to defend body against microbes that can cause disease, like so. Next, we have receptor proteins. Receptor proteins may react to or move a signal from one side of the cell membrane to the other. And of course, we have enzymes. These are biological catalysts made by all living cells. They speed up cellular reactions and are unchanged in the process. As a result, they can be used again. The shape of the active site of a particular enzyme molecule is complementary to a specific substrate. And if we now look at a typical enzyme reaction, the substrate is the molecule acted upon by the enzyme, and the product or products are the molecules produced by the action of the enzyme. There are different types of enzyme reaction. Degradation reactions are when complex substrates are broken down into simpler products, and synthesis reactions are where simple substrates are brought together to form complex products. And these are some examples of enzyme reactions. Many of the enzyme reactions that we learn about are degradation reactions. The one in particular that is a synthesis reaction is the action of phosphorylase producing starch from glucose. All enzymes have optimum conditions. The optimum condition is the value at which the enzyme is most active. Enzymes have optimum temperatures and an optimum pH. Optimum temperature. This is the temperature at which the enzyme is most active. In this case, it is about 40 degrees C. Now, if you have a look at the effect of pH on other enzymes, pepsin works well in low pHs. Most enzymes in the body work around a neutral pH. There's one enzyme in particular, alkaline phosphatase, that works best at a higher pH. So the optimum pHs, the pHs at which those enzymes are most active, are like so. If we have a look at the effects of high temperature on an enzyme reaction, normally the active site of the enzyme fits exactly with the substrate, the enzyme reaction occurs and the products are made. However, if we take that enzyme and place it into boiling water, for example, the shape of the enzyme changes. The active site is no longer complementary to the substrate, and that change in the structure of the enzyme is now permanent, and we say that the enzyme is denatured. It will no longer work.